My name is John Duncan from the Queen Square Institute of Neurology, part of UCL London and the National Hospital for Neurology and Neurosurgery. Myself and my colleagues Menika Sidhu and Vijay Vakaria have had a big interest in the use of imaging to guide epilepsy surgery. And in this chapter, we describe preoperative functional localization with fMRI and diffusion imaging and tractography. Epilepsy surgery can be curative in up to 80% of people with focal epilepsy, but this carries significant risks of causing postoperative morbidity. And we'll show in this chapter that identifying and visualizing eloquent cerebral areas and critical white matter tracts is important for mitigating the collateral damage from epilepsy surgery. Functional MRI can lateralize and broadly localize eloquent functions of language, memory, motor, sensation, and vision, and Menika will go through this in detail shortly. Tractography, derived from diffusion MRI, can infer the location of major white matter tracts, particularly optic radiation relevant for temporal lobe surgery, corticospinal tracts, and also the language-related tracts of arcuate fasciculus and inferior fronto-occipital fasciculus. A very important caveat is that data must be acquired very carefully and interpreted cautiously, and can be a very useful adjunct to precise neurosurgical navigation to avert the causation of deficit.
The use of cognitive fMRI in epilepsy surgery is really threefold. Firstly, to lateralize and localize language and memory functions preoperatively, in the prediction of memory and language outcome after surgery, and finally to describe the postoperative plasticity in these cognitive networks. In most surgical centers, language fMRI is used instead of intracarotid amylobarbital testing to lateralize language functions. The benefits of language fMRI over intracarotid amylobarbital testing is that firstly, it is a non-invasive procedure. Next, a larger repertoire of tests can be performed in a single setting. And finally, repeated testing is possible with minimal risk to the participant. Verbal memory functional MRI has been shown to be the strongest independent predictor of verbal memory outcome after anterior temporal lobe resection. And this was compared to standard neuropsychological measures such as preoperative memory as well as language lateralization. Using an LI of 0.5, which means more left lateralized activations within a frontotemporal mask, memory fMRI predicted verbal memory outcome with 87.5% sensitivity and 80% specificity. Using a very similar individual prediction model, language lateralization greater than 0.4 within a large temporal mask predicted naming decline with 100% sensitivity and 92% specificity after anterior temporal lobe resection. Practice guidelines from the AAN of the use of fMRI in the evaluation of people with epilepsy suggest that language fMRI may be used instead of intracarotid amylobarbital testing for language lateralization in temporal and extratemporal epilepsy. Memory fMRI can be used instead of IAP to lateralize memory functions, but only in temporal lobe epilepsy. Verbal memory fMRI should be used to predict verbal memory outcome, and visual memory fMRI could be used to predict visuospatial memory. These guidelines were published in 2017 and likely to evolve further.
When interpreting tractography results in a clinical context, three main important concepts should be borne in mind. The first is that diffusion imaging measures the movement of water molecules and, through this, enables inferences to be drawn regarding the directionality of fibre bundles within the white matter. The second is that an algorithm must be applied to calculate the diffusion orientation distribution function for each voxel. Different algorithms return different results, with more sophisticated algorithms being able to resolve crossing and kissing fibre populations within the same voxels. Finally, streamline propagation through different tracking algorithms provides a mathematical estimate of water movement between adjacent voxels. The streamline density reflects the probability of water diffusion in a particular direction and is not a quantification of the number of axons. Free water molecules move equally in all directions and is termed isotropic movement. When the movement of water is restricted in a specific direction compared to another direction, this is called anisotropic movement. The extent to which water molecules within a voxel show anisotropic movement is quantified as the fractional anisotropy, Fa. Regions of the brain in which the axons are uniformly aligned, such as the corpus callosum, return high Fa values as movement of the water molecules are restricted to the direction of the axons. Correspondingly, free water movement, such as in the lateral ventricles, return low Fa values. The application of magnetic gradients at different directions allows the directionality of water molecules within each voxel to be quantified. In the simplest form, water molecule movement can be represented mathematically as a tensor with the principal eigenvector, lambda1, assigned to the direction with greatest movement. This represents the longitudinal diffusivity. The two orthogonal directions assigned as lambda 2 and 3 represent the radial diffusivity. It is common to visualize these as a diffusion-encoded color map. Voxels, in which the longitudinal diffusivity is in the cradiocaudal axis, are colored blue, anterior-posterior axis, green, and the left-right axis, red. Voxels with multiple fibre populations that are not aligned are not well represented, and the diffusion-encoded colour map may be misleading. More sophisticated approaches have been employed to derive the fibre orientation distribution function, which allow multiple fibre populations within a voxel to be differentiated. Tractography utilizes tracking algorithms to assign connections between different voxels. The user assigns a seed region from which streamline propagation begins. Fibre assignment through continuous tracking, FACT, was the first method to be used and is a deterministic algorithm. This means that the direction of the streamline propagation follows the principal eigenvector, lambda 1, and there can be no deviation from this. This is likely to represent the tract well. That is to say that there will be a high number of true positives, but will be an underestimation of the tract as a whole, so there will be a large number of false negatives. This is useful for indications where a high level of precision is required, such as targeting a specific tract with a deep brain stimulation electrode, for example. Probabilistic algorithms model the inherent uncertainty of having multiple different fibre bundle populations within the same voxel, and each voxel is, therefore, connected to the adjacent voxels based on a probability distribution derived from the diffusion orientation distribution function. This method is most useful for indications where a tract is to be avoided, as the reconstruction is likely to be an overestimate. That is to say, it will contain more false positives. The streamlines are also refined through user-applied restrictions, such as defining inclusion and exclusion regions of interest, where streamlines passing through the region are retained or removed, respectively. Within the same patient, different tractography results are achieved based on the scanning parameters, such as the number of gradients, which may be single or multi-shell, the gradient strength, represented as the B-values, and the number of directions acquired. Additionally, the variability in the method used to calculate the diffusion orientation distribution function, positions of the seeds, inclusion and exclusion regions of interest, and tracking algorithm all affect the representations of the tracts. The reliability of the tract reconstruction is based on an estimation of the known anatomical course of the tract, as there is no in vivo gold standard for comparison. Efforts are ongoing to standardize tractography through the implementation of automatically derived regions of interest, and these have been shown to be comparable to expert human reconstructions, but caution is required when utilizing tract reconstructions intraoperatively, as it does not provide information regarding function. Confirmative direct electrical stimulation is recommended when employing tractography, as this represents the gold standard for intraoperative functional mapping. Within epilepsy surgery, the optic radiation, corticospinal tract, and language tracts are most commonly reconstructed. Preoperative probabilistic tractography of the optic radiation has been shown to be able to predict visual field deficits in patients undergoing temporal lobe surgery. Through the integration of neuronavigation systems with the operative microscope, tractography results can be visualized directly as overlays within the operative field, and have subsequently been applied prospectively to guide anteromesial temporal lobe resections. These corticospinal tracts can be reconstructed using seeds derived from fMRI data, such as during finger tapping and lip puckering tasks, or from anatomically placed seeds. Within epilepsy surgery, the epileptogenic zone may be larger than the visualized lesion or area of recorded electrographic seizure onset following invasive recordings. Tract reconstructions may be used to plan maximal resection volumes whilst also preventing neurological deficit. Whole brain connectomes can be derived utilizing tractography, and network analyses can then be performed at local and global scales through the application of graft theory metrics. This opens the potential for minimally invasive techniques such as radiofrequency ablation and laser interstitial thermal therapy to disrupt the seizure network and prevent seizures from spreading.